Hi everyone! In this video, we will see how the ColE1 plasmid regulates its copy numbers within a cell. Here is a genetic map of the ColE1 plasmid, which is figure 4.5 taken from the 4th edition of Snyder's Molecular Genetics of Bacteria. On it, you can see the origin of replication, as designated by ORIV. This is where DNA polymerase will bind to replicate the plasmid. The promoter for RNA2 is designated by pRNA2. The region designated by INC will produce a secondary RNA transcript, RNA1. It is also important to note that the region designated by ROP encodes for an important regulatory protein ROP that we will see later on. Let's start by looking at how the ColE1 plasmid replicates normally. In this picture, the plasmid DNA is represented by two blue lines. However, naturally in the cell, it would be circularized. To begin the process of replication, RNA polymerase binds to the RNA2 promoter and creates the RNA2 transcript. From there, the RNA transcript moves to the origin of replication, forming an RNA-DNA hybrid. Then the endonuclease RNase H comes in and cleaves the 3' end of the RNA. The cleavage by RNase H leaves a free 3' hydroxyl group. This free hydroxyl serves as the primer for DNA polymerase I to come in and replicate the plasmid DNA. However, we know that the cell does not want to make an excess of plasmids. Therefore, what happens to this process when there is a sufficient copy number of ColE1? As we know from the genetic map, ColE1 does not just have the promoter for the RNA2 transcript. It also has the promoter for the opposite RNA1 transcript and the ROP regulatory protein. ROP, as pictured in purple, is primarily found in the dimer form. When both transcripts are made, they tend to associate due to the complementarity of their base pairing. This is stabilized by the ROP dimer. However, the dimer is not required. This loose association of the two RNA transcripts is called the kissing complex. Often, the RNA in the kissing complex associate tighter, forming what is known as the hugging complex. When they are tightly bound, RNA2 undergoes some conformational change so that it cannot form the RNA-DNA hybrid and therefore is not cleaved by RNase H. Without the free hydroxyl, DNA polymerase I cannot bind and therefore there is no replication of the plasmid. So let's review. Remember, in normal replication, only the RNA2 transcript is produced. It is then cleaved by RNase H and serves as the primer for DNA polymerase I. When there is a sufficient copy number, RNA1 and ROP will also be transcribed. This will form the kissing complex that prevents DNA polymerase from binding, and therefore no replication can occur. Thanks for watching, everyone, and I hope this was helpful.